Yeah, so as Marissa said, I'm coming from Glasgow, not literally, but near enough. I'm not an acoustic expert, but I am a spatial epidemiologist and a disease ecologist. So hopefully this is going to act as a bit of a sort of inspiration or a signposting to other ways that you guys could use your expertise, and then how some of my colleagues are starting to use passive acoustic monitoring in this field of study. So the talk, I'm going to give an intro to infectious diseases and zoonotic diseases, which is kind of more on the sort of in-depth technical side. We'll have an acoustic break in the middle, and my colleague Maz is going to be talking about how they're using acoustic monitoring in Borneo. And then I will bring it back round to how this all ties together. So, oh, no. Yes, okay. So zoonotic infectious diseases and emerging infectious diseases pose a severe global public health threat. We're all fairly familiar with that now, but just so that everyone is on the same page, zoonotic spillover is the transmission of pathogens from vertebrate animals to humans, and this might include vector-borne diseases as well. So zoonosis is any disease or infection that is naturally transmissible from a vertebrate to a human in its ecosystem, in its environment. And there are over 200 known types of zoonosis, each with animal reservoirs, and importantly, that means that each has a specific etiology and ecology of transmission. So I know a lot of people here are probably ecologists. And high-profile examples of this would be Ebola virus or pathogenic influenza or MERS virus, as well as more localized or endemic transmission like Nipah virus or leptospirosis. And vector-borne diseases such as yellow fever or simian malaria. And emerging infectious diseases are widely linked to land cover and land use changes, so from as early as 2015. And this includes agriculture, plantations, urbanization, deforestation, and forest fragmentation, where the forest is degraded. And generally, evidence continues to mount that human alterations to landscapes and habitats is a crucial factor in spillover of disease. So impacting the prevalence of pathogens in reservoir hosts, linking to the incidence in vulnerable populations, and generally changing the ways in which humans, wildlife, and pathogens interface. So this is the intense bit. Likelihood of spillover is the key thing here, and it's determined by the interaction of several factors, including pathogen dynamics in the reservoir, exposure of susceptible host species. And if we think a little bit about those cascading mechanisms, the idea of pathogen pressure is partly determined by ecological dynamics, such as distribution and abundance of the reservoir species and the vectors, the wildlife species, and where the pathogen is sustained in circulation. So this means that the behavior and movement of humans and vectors and wildlife determine the likelihood of exposure. So we have all of these epidemiological determinants interacting with ecological processes. And in order for these cross-species transmissions to occur, wildlife reservoirs, vectors, and humans must have the opportunity to interact in space and in time and within certain environmental contexts. So we're already starting to think about the distribution and the abundance of certain reservoirs and how they interact with other species in the same context. And with so many moving parts to this, so to speak, the determinants and the dynamics of zoonotic and vector-borne diseases are really complex, and understanding these requires research and data that is cross-disciplinary, integrating wildlife ecology, landscape and spatial ecology, human epidemiology, and behavioral studies. And to understand these opportunities or windows for spillover, depicted here as sort of holes in each layer, there are some basic core ecological questions that we can ask. So this includes which species are present in an area, where and how key hosts move in space, when species are present, and by this I'm referring both to reservoir species and to humans, so the susceptible hosts, and thus whether there is spatial and temporal overlap between these compartments. So a break from information, some maps. If we start to think about where we need to be asking these key questions, here we have a map of predicted risk distribution for zoonotic emerging infectious disease events. It's a little out of date now, but the principle stands. And the headline is that the risk is mostly elevated in forested tropical regions that experience land use change in particular, and in areas where biodiversity is really high, as well as areas with high urban density and animal trade and so on. So you can visualize that further by overlaying maps of forest gain and forest loss, and you can see that where there are hotspots for zoonotic spillover on the previous map correspond to areas with extensive landscape change in recent decades, particularly Central America, West Africa, and East and Southeast Asia. And I saw on the intro to where you're coming from that lots of people are coming from Southeast Asia. And this maps further onto gaps in biodiversity data. So I know a lot of people here, again, are biodiversity oriented. And zoonotic disease risks do correlate to land use and land cover and with wildlife biodiversity. And here, when we look at where biodiversity is monitored relative to changes in land cover, we can see that most of the monitoring occurs in Europe and North America, as well as South and East Africa and Australia. But there are big gaps in monitoring in South America, in West Africa, and East and Southeast Asia, which are all regions experiencing extensive landscape change and are hotspots for zoonotic spillover. So if we're thinking how you might monitor, this is all bread and butter to you guys, but thinking about how you can monitor wildlife, trapping, tagging, GPS collars, using camera traps, and more recently, particularly with colleagues of mine, we have expansion in the use of Earth observation methods. So that's drone imagery, which can be fitted, for example, with thermal cameras to track megafauna and things like this. And these field methods are incredibly valuable, and they do provide important insight to small-scale social and ecological processes that underlie disease risk. And we do use all of these, but they have their limitations. They are generally time and resource intensive. Visual remote sensing technologies are directional and limited in areas with dense canopy cover, like tropical forests, or indeed tropical regions. And given that zoonotic disease transmission and risks are most pronounced in those landscapes, often with challenging access and terrain, these limitations really are quite limiting when trying to understand ecological dynamics of infectious disease risk. And classical methods aren't always suitable for understanding disease dynamics at scale or in these forested environments, which is why I'm here talking to you guys. So acoustic monitoring is, I mean, you guys know all this. It's a process of recording sound, for example, animal vocalizations, anthropogenic noise, or biotic and abiotic sound. And bioacoustics can be analysed to understand a great deal about the environment, including presence and behaviour of species, about the health of the ecosystem. And passive acoustic monitoring is obviously the specific use case of this acoustic technology, where sensors can be programmed and deployed for continuous autonomous acoustic surveys. So it requires, relative to other wildlife tracking, relatively few resources to collect a wealth of ecological data that can be applied to infectious disease in this case. So this all culminated in a review paper and a trends paper looking at the applications and advances in acoustic monitoring for use cases in infectious disease epidemiology. And as we've shown, there are big gaps in our understanding of the mechanisms underlying zoonotic disease dynamics. And I can talk a bit more about that as well, but particularly in areas experiencing land use change and landscape change. And there are shortcomings in classical methods for answering some of these core ecological, epidemiological questions in tropical context and at scale. So this paper aimed to highlight the opportunity and utility of acoustic sensing technology in answering some of these questions related to zoonotic disease risk and human epidemiology. So that brings me to the sensor project. And in a moment, I'm going to hand over to my colleague Maz, who's on the call, to talk about what they've been doing in Malaysia. But this was it's a project based in Malaysian Borneo, and it's a longitudinal observational cohort study in human participants of infection and exposure to zoonotic and vector-borne disease risk across a land use gradient. So we have a deforestation system and we have a reforestation system, which is where Maz is based. So it's a socio-ecological systems approach. And one aspect of that is using audio moths to start to understand some of these dynamics, which I'll talk more about later, but Maz is on a generator time frame, so I'm going to hand over to Maz, if she's there. I'm hoping she still is. She might have.
Can you see me? Hi.
Yeah. Hi.
Maybe you can hear me, maybe you don't see me because I am in the middle of Bornean jungle, so our Wi-Fi is a bit bad. But do you hear me?
We can hear
Hello?
you. Yeah,
Yeah.
we can hear you.
Okay. All right. So yeah, we've been collaborating with Emilia and the other colleagues on these bioacoustics. But
All good. If you also want to maybe freeze your camera, turn off your camera just so that the audio comes in even smoother. We're mostly getting smooth audio, but just to double check.
Yeah.
Yeah.
Okay. All right. Is it better?
Yep, go ahead.
Yep. Okay. So we have been collaborating on this project. So this is a case study in the Kinabatangan area, floodplain in Borneo. And we've been using bioacoustics actually in our restoration plots. So what we are doing in Borneo is we are restoring the forest and we are monitoring multi-taxa animals in Borneo. So we are doing a lot of monitoring on frogs, on birds. We're doing it on small mammals, dung beetles, and we're doing we this actually to our project enforcement, so wildlife trading. So we got this to help us with detecting like machine or like gun in the forest from hunters, poachers, and also the illegal logging. And then we decided to also use these devices to monitor our restoration plots. So we deployed them in our active planting site where we newly opened the site for planting. And we also deploy them in the restored sites where we have sites we have planted since like 20 years ago. And also in the natural forest, it's just to compare what kind of species we find in these different habitats and different forests. Design, we deploy the audio modes every month for 10 days, and it's recording for 1 minute every 5 minutes, and it's along the Kinabatangan River. So Kinabatangan River, it's like 500 something kilometer long, and we deployed our devices along the river. And as you can see, we also put the device in the cage because there's a lot of risks from animals and damage from animals, especially macaques, because they sometimes just steal the audio moth, and sometimes squirrels, they just go and eat the device. So we really need to secure them. And also because our our area is very it's in the floodplain, so it gets flooded frequently. So we need to put it like higher, and I don't know, it's maybe after this one, but so for this for the configuration, we normally just use 48 for sampling rate, and medium for gain, and recording for 1 minute every 5 minutes, 10 days deployed in the sites. And okay. And so we are using the Arbimon website. We are also collaborating with RFCX. So we use these sites, and I think you guys are more familiar with all these things because you guys are doing, bioacoustics. So yeah, mostly, uploading all our recorders on this website. And yeah. So mostly this is actually RFCX that we are collaborating with, and they are helping us with all the analysis as well, and all this pattern matching, where they match all these sounds that we got from our sites. But Marconi is not able to join today, but he's mostly the one who's working on all this analysis. So yeah, that's the project that we have in Borneo at the moment. So mostly related to forest restoration and monitoring of biodiversity.
Perfect. Thank you, Maz. Yeah, so Maz and DGFC in Sabah are working with Rainforest Connection a lot. As Maz said, Marconi couldn't join us, and he's our kind of technical lead on the paper. So any technical questions, I don't think you guys would have any, but that's where they should go. And I don't know whether these will play, but as a sonic interlude, I do have some examples of the clips from Malaysia, so I'll see if they'll play. Whether they're playing. Marissa, can you give me a thumbs up?
No, the sound isn't playing through.
Sound is not playing. Okay, I'll skip them. But yeah, these are some examples of what we're collecting from DGFC for infectious disease, and I'll now go on to a case study to sort of exemplify that. Okay, so this is all Marconi's kind of pipeline of analysis. Again, I'm sure you guys know this better than we do, but I will flick through the slides. If you want the slides or to get in touch with me or Marconi, please do. But yeah, this is the workflow for how the data is being analyzed. Examples of primate identification using pattern matching. So we're looking particularly for macaques, long-tail macaques. I'll go into why in due course, and also soundscape analysis. So looking for whether we can detect changes in the quality of the forest. So with the reforestation plots being between 20 to 2 years old, seeing whether there is any difference in the biodiversity and kind of wildlife assemblages, and whether that's detectable in the soundscape analysis. And again, this is the technical side of the analysis. I'm gonna do, yeah. So case study. Particularly pertinent to those from Southeast Asia, I'm going to be talking about zoonotic malaria, or Plasmodium knowlesi. So Southeast Asia has seen a near total decline in human malarias, so the malaria we're familiar with from particularly sub-Saharan Africa. But at the same time, there's been this massive spike in zoonotic malaria. So it's associated with a wildlife reservoir in non-human primates, and it's transmitted by particular mosquitoes from primates to humans. and it's now the main source of malaria infections in Malaysia. And since human Knowles-eye infections are a result of spillover events from ecosystems into human populations, we need a more comprehensive understanding of transmission dynamics at these interfaces. And this is again what we saw earlier, that that on a very broad scale, PCR testing of Knowles-eye cases, you can see hotspots in Malaysian Borneo as well as Thailand, and when you then overlay a map of tree cover and forest loss, you can see there is a clear spatial overlap between these areas experiencing high rates of deforestation or forest fragmentation, and high incidence of human cases. So it suggests a link between the two. And this is a bit about the sort of epidemiological background of Knowles-eye. So it has a transmission cycle that intersects with wildlife behavioral dynamics and vector biology and human movement, as well as land use, and it's all embedded in health systems. But the effect that landscape change has on disease dynamics is complex and it varies across multiple spatial scales. But looking at human Knowles-eye, they found a strong association between village level Knowles-eye incidence and localized forest cover and historic forest loss. This is just sort of the kind of data backing up the association. And with regard to vector ecology and some of the other facets here as well, it's been shown that forest fragmentation favors vector breeding sites and abundance for known vector species, and landscape features also impact wildlife in this disease system. So localized deforestation changes macaque movement and ranging behavior, and forest fragmentation have been shown to drive higher parasite loads in the macaque reservoirs, so in non-human primates. But what we don't yet understand is these spillover mechanisms of the mechanisms of where and when humans, vectors, and macaque reservoirs are interacting within these fragmented landscapes. And these are the methods that we're kind of using so far. So Earth observation for the landscape, GPS tracking, and more kind of public health serology. And for the macaques, Earth observation data, GPS tracking, and field biology. And this is kind of a great example of where acoustic monitoring can kind of address a gap. So starting to think about, does ecosystem health impact disease risk? Where are humans, mosquitoes, and macaques interacting? And when humans, mosquitoes, and macaques are interacting. So this is the key figure from the paper, and it's an example so the paper was designed for epidemiologists mainly to start thinking about how passive acoustic monitoring could be integrated into infectious disease monitoring and surveillance systems. But the same is true for acoustic monitoring audience, to think about the problems that you're already working on and the techniques and the kind of sampling designs and the complexity and the statistics to try and analyze all this data, how that can be retrofitted to questions for epidemiology, for epidemiologists. So here, examples of occupancy, so where macaques are, spatial estimation. This is the pictorial reference to Nipah virus, which arose from a spatial overlap between fruit bats and pig farms, mosquitoes at a certain hour, behavioral traits, sort of acoustic signatures of breeding seasons, which tend to peak with Marburg risk, and more generally kind of ecosystem health, sort of soundscape metrics for ecosystem health. So to link this back to the Plasmodium Knowles-eye example, as part of the paper, we kind of mocked up some of these ideas of how you could apply acoustic monitoring to Plasmodium Knowles-eye. So here we can use acoustic sensing to collect fine-scale data for macaque movement, for example, across space and time. This is from the Kinabatangan, where Maz is. And we, using cloud-based computing and pattern matching approaches in Arbonne, acoustic data was analysed to detect long-tailed macaque presence across different habitats in areas that were known to be endemic for Knowles-eye. And here the map of detection points for the long-tailed macaque, blue points, pink points indicating locations with acoustic sensors where they were detected, so only 4 out of 27 sites. Macaques aren't the most soniferous species, so there's a little bit of the sort of typical challenges there. In time, temporal estimation can also provide valuable information for risk patterns. So for mosquito-borne diseases such as malaria, detection of human movement at critical times could be used to indicate heightened transmission risk, and could then be used to prioritize vector control, particularly in areas that are nearing elimination. So here, increased human activity during key mosquito biting times, which was that was parameterized using entomological data, that overlapped could suggest something about the malaria receptivity of a given area. So that's essentially the times of greatest risk for humans that live and work in the area. So by comparing detection frequency of human chainsaw use in a high logging area against bite rate for the main malaria vector, we can see high risk times for malaria transmission, and in this hypothetical population would be early evenings. So you could use that to inform public health interventions. And for the context, in understanding the ecological context of disease transmission risk, soundscape analysis could be useful to assess and compare animal richness through time and space. And that could then be valuable to understanding altered disease transmission potential, linked to changes in biodiversity or overall declines in ecosystem health. So for instance, species richness of insects, birds and primates are strongly and positively correlated with the acoustic space used, and this could then be related back to kind of spillover potential, particularly in fragmented habitats. So that was a bit of a whistle-stop tour. These are the current projects that my collaborators have on. So in Guinea, in the centre, there's a project looking at Marburg, so they're mapping human and bat reservoir distributions within case study villages, and using acoustic data in conjunction with bat roost surveys and human GPS studies. The Gambia, same for Rift Valley fever, which is more of a livestock disease, malaria in Peru, and the Kinibatang in Borneo, again looking at overall biodiversity in the reforestation plots, but also how this translates to gnawseye risk. And the bottom line is that this technology is just starting to be applied to these questions, to the epidemiological scenarios, and it's in a variety of contexts and settings. And I don't need to remind you of this, but the benefit is it's non-invasive to collect data that is spatially and temporally explicit, which particularly for epidemiology is really powerful and very, very important, locally and at scale, without the logistical intensity that classic fields ecology methods require. Mainly, it has the potential to complement existing methods. So detailed data to complement existing intensive data and addressing logistical and conceptual shortcomings. So combining acoustic survey data with data that is even higher intensity but sparse or expensive, it can strengthen existing data sets. There are obviously limitations. It definitely does produce, it definitely is a new opportunity for epidemiology, and it's really exciting. But it is obviously limited to disease hosts and vectors that produce sound, and for novel areas or species that aren't typically the subject of biodiversity or conservation surveys, but are a risk for disease. The lack of training data might be an issue, might limit the usability, and might impair the sort of use of supervised learning algorithms and species detection and monitoring. And yeah, general consideration, again, more for epidemiologists, is it offers a valuable addition, but it doesn't replace existing methods, so it's best used in conjunction. And as an emerging application, you need to have a think about how this is going to be practically deployed and evaluated, and how analysis pathways can be adapted for disease-specific applications. So that's going to require specific workflows for integrating acoustic data into research and surveillance. But yeah, overall, acoustic monitoring provides really exciting opportunities for the field of human epidemiology, and offers a new tool to understand spatiotemporal risk for emerging zoonotic infectious diseases. And just as I don't know how I'm doing for time, but just as a kind of reminder or bringing it back to the general context, as Marissa said at the beginning, acoustic monitoring is most generally used in ecology, biodiversity, and conservation. But this is just a figure I nicked from a recent review, a recent Lancet review that was looking at the linkages, the bi or tri-directional linkages between climate change, biodiversity, and infectious disease. So there are some huge wins to be gained from looking at where these global pressures intersect. And yeah, I think that kind of speaks to where acoustic monitoring could provide real value. Just as a kind of, again, from the same paper, case studies linking all three, so not just disease and biodiversity, but also disease, biodiversity, and climate change. For those who are kind of seal and whale people, examples being phocine distemper, which is really closely related to rabies in humans, and also, I mean, it's well known for kind of diseases that affect amphibians, but also this last example in bee, sort of shifts in the seasons, Ebola outbreak risk, and primate community assemblages is another example where you could tie in acoustic monitoring to kind of study some of these interactions at a fine scale. And as I said, we work with Rainforest Connection, and this is their home screen, because, as you know, they use acoustic monitoring to detect logging as one of their case uses. So the most impactful way to stop climate change is to save rainforests, and this is from the Pandemic Commission on Stopping Spillover, How to Stop the Next Pandemic, Stop Deforestation. So there is obviously some intersection here. And if you need even more examples of why this sort of why disease ecology and why ecology and biodiversity is so essential, the Lancet are now funding this new, or have commissioned this. It's a commission specifically looking at spillover, so that intersection between wildlife and environment and ecology and disease, with a view to reducing pandemic threats. There is a lot of traction in that area, and one facet of that is acoustic monitoring for wildlife hosts of zoonotic infectious diseases. So I would like to acknowledge everyone that helped with this, Maz from DGFC and all the other institutions, and Kim from the NUS, whose project this was. This is all of us. At this point, I would normally open the floor for questions, and I can do that. But when I emailed Ben warning him that I was not an acoustic expert, he suggested that I should use this opportunity to a group of bioacousticians to ask, how would you approach some of these questions and problems? So maybe more of an open forum, anything that you think is exciting or problematic or challenging, or where your existing research could lend itself to epidemiology. So thank you for listening, and I hope that was interesting. But thank you for inviting me to talk.
Wonderful. Thank you so much, Emilia, to you and your team. It's really fun to use BioacousTalks as a forum to kind of put our heads together and think of ways to expand acoustics as a skill set to answer questions in lots of different areas. I want to open up the floor. I mean, there's lots of good material in there. Anyone want to chime in either with a response to the open forum question about how to approach some of these questions or problems, any comments, doesn't necessarily need to be a question, or anything that you're curious about. Feel free to either raise your hand and unmute, or you can throw a question in the chat too if it makes you more comfortable, and I'm happy to read it out loud.
Hi, I'm Yan Yi. Thanks for the talk. I actually saw your paper last year, and I know Ashraf as well from your co-authors list. And I actually got inspired by this paper, and I'm thinking of starting a project on using bioacoustics to look at disease spillover, and I have collaborators who are epidemiologists and virus what do you call those? They can detect viruses from samples. So I was wondering if bioacoustics could be used to detect some of the behavioral aspects of animals where, like for example, in bats, when they are stressed, they are more likely to shed virus. But I wasn't sure if we could detect that sort of thing from acoustics, and I wonder if you have any ideas on that and how we can actually relate their sound with whether they have disease or not, something like that. Yeah, thanks.
Thank you. That's really nice to hear that you were inspired by the paper. And sounds like a really, really exciting project. I think actually the last time we presented this, we had the same question, whether you actually could detect any behavioral signatures that would correlate to sort of viral shedding. I don't know whether you can or not. I think that certainly for some, for example, the Marburg. So the Marburg example, the shedding is higher during maternal roosting. So they have seasonal breeding, which you can detect in acoustic signatures, and that is a correlate of shedding for things like stress. I'm not sure. I mean, someone else here might know better about acoustic detection in bats. But yeah, I mean, it'd be really a fascinating project.
Yeah, thank you. We're actually thinking of conducting this project in a city where we might be able to get data from hospitals of how how many patients got got infected, and then relate that to the distribution of bats or any other animals. So yeah, actually I'll be interested to talk to you after this. Maybe you can set up a meeting through Ben, or
send me an email.
we can talk more about this. I don't want to dominate the forum. But thank you. Yeah.
Wonderful. That's what BioacousTalks is all about. We have another question in the chat from Anna. I'm currently absolutely amazing talk. Thank you. I'm currently developing a project employing passive acoustic monitoring to identify avian influenza affected sites based on infected individual sounds. You can actually tell who's sick. Very cool. I was wondering whether you are employing direct disease detection, oh my God, in any way.
That sounds like an absolutely fascinating project. In the systems that we're using acoustic monitoring, I don't think we are using any direct disease detection. But I have a lot of colleagues who also work in avian influenza, and particularly in some of the really vulnerable gannet populations that have been decimated by influenza. So I mean, that's really exciting that you can tell who's sick in the populations. Yeah, I'd love to hear more about that.
Anybody else wanting to chime in either over chat or verbally? In the meantime, I have a question for you, Emilia. I tend to work in marine ecosystems, and I'm curious if you've seen, I'm relatively new to this approach and application. I'm wondering if you've seen in your line of work any applications toward using PAM for the transmission effects also in ocean environments.
Yeah. So this is not specific. I can answer to the disease in marine and where you could use PAM rather than existing examples, but I have a colleague who is doing her she's doing her PhD on rabies virus in Peru, and they have rabies spillover into the seal populations and sea lion populations along the coast because they have cave roosting bats that are feeding on the sea mammals. So that is, I mean, that's a great example of kind of terrestrial ocean interfacing, and specifically a disease system. So I don't see why passive acoustic monitoring wouldn't be advantageous in that system.
Sure.
And I think that's probably the best example. Otherwise, I have colleagues who are looking at kind of viral transmission between cetaceans, but mostly on postmortem. So you probably wouldn't use acoustic monitoring there. But yeah, I mean, if you can, I mean, I'm not a whale person or an ocean person, but if you can detect signatures of disease or kind of behavior that correlates to disease, I don't see why not.
Yeah, that's a very interesting food for thought. I hadn't known about the sea lions and the rabies.
Yeah, and I mean also the aside from her project, the kind of mass strandings and die-offs in seals and other cetaceans from avian influenza, in South America. There may well be some acoustic signatures to be found there as well.
Mm-hmm. And we have another question from Tanisha. Were you able to get some audio recordings of big wildcats, for example, pumas? If yes, then what was the minimum range or distance till the audio moth could record the sound?
That is probably one for Maz. Whether you're still online.
Sorry, what was the question?
If you were able to get audio recordings of big wildcats, and if so, how far could you detect them?
For this one, I don't think we have had cats, but we tried. We tried to deploy some of the devices in the oil palm plantation because one of my colleagues is working on cats in the plantation, wild cats, but we didn't really hear any in the recording. So yeah, so I cannot really answer. I mean the range, yeah, of the detection. But you definitely can do it on cats as long as they vocalize. Yeah.
What would you say would be the main predator, what is the most apex predator group that you were detecting in your ecosystem?
Me?
Yeah.
Actually, because we didn't, for the pilot study, we didn't really deploy them like very long in the site. But mostly we were just trying to analyze birds and
Makes sense.
primates mostly. Yeah, primates. So we didn't really get sounds from, like the main predators like the clouded leopard as well, because they They are also considered as, predators. Yeah, so we didn't get clouded leopard in there. So mostly like primates, gibbons, proboscis monkeys, macaques, and birds mostly, actually. Yeah.
Very cool. The floor is open again. Any questions people want to unmute to ask, or anything they want to toss in the chat? Ooh, go for it.
Okay, hi, hello. I'm so sorry I cannot open. The in here is already night and my baby's sleeping, so I can open video.
Oh, no worries.
Yeah,
We're glad you could
I
join.
just yeah, okay. I'm Miska from Indonesia, and I'm just still very new in the bioacoustic, and I would like to know. This is in my opinion. I try to using my camera traps, and then I get the acoustic, the sound from the camera traps. And after that, I'm doing what you call, are you using audio mode, right? I'm so sorry, you're using RB mode and for your analysis. However, in here, I also, after I get audio from camera traps, I try to connect this kind of sound from camera traps and then using RB mode. And one or two, we can hear the sound, and maybe I don't know because I'm still very new in Arbimon also, so I'm still learning. So I can detect one or two or three insect or sometimes a bird, and because I don't know how to use the template, and the other I still don't have anything. Did you ever have using, because you before have a PP in your slide, you talk about the camera trap, etc. Do you ever try to use the camera trap and then use the sound from camera trap and using it to bioacoustic too? Because in here I'm so difficult to find I mean in my places I'm difficult to find any kind of device in bioacoustic, so we're still using camera trap and I'm just trying to figure out for this one. Thank you so much. Sorry for the long questions.
It's okay. I'm sorry I can't help more. I've never used the acoustic from camera traps. I don't know whether Maz you have. I know you use them at DGFC, or anyone else on the call, but personally, no, sorry.
Yeah, I've never used sounds from camera traps as well. So mostly from the audio mode. Yeah.
In that case, has anyone in this room used the sound from camera traps that wants to speak up about their experience? We welcome all thoughts.
Hi, everyone. Ian from Virginia Tech. For my dissertation research, I used camera traps and passive acoustics together. And when I wanted to look through the camera trap data acoustically, it was possible, but the quality was a lot lower. So that's just something to keep in mind.
Ian, if you don't mind me following up on that, by quality, do you mean like you were getting bad signal to noise? Do you mean that like you just couldn't record as much? Like in what ways, like was it lower quality?
The frequency is mainly lower frequencies. Because with the passive acoustic, I think I was at 48 hertz. And it was a high-end camera trap, but I think it was still it might have been only 4. I can't remember.
Right.
But they pair really well together just because you can identify events and camera traps and the acoustics and match them.
So if you have lower frequency species, that might be a ideal solution or scenario.
Yeah, that sounds right.
Cool. Yeah, and I see that you also just threw a question in the chat and you have your hand raised, so go for it.
Yeah, just wanted to add a little bit more about the camera trap sound as well, because I did do a small analysis on that. I do realize that if the camera trap is really close to your sound of interest, like if you're pointing at a nest or a roosting site, or it's on a fruiting tree or something, it might have better quality. But I haven't tried it on a range of animals. So yeah, just wanted to add on that. And I have a question on the chat. So what would it take to create an early warning system of disease risk for different animals? Yeah, I'd like to hear more about that.
Thanks. Yeah, I mean, that's a really great question. That's kind of the question. I think, I mean, obviously I can't speak totally to the acoustic monitoring side of that, but I think that all of the current challenges in terms of abundance estimation and the kind of reducing the biases and the sort of spatial what's it called, statistical non-independence of similar sounds, you know, that is all part of the challenge, but specifically for disease systems, part of the challenge is understanding exactly what the ecology and the etiology of the spillover is. So it could be, for example, MIPA and spillover from specific fruit bats into pig farms. That's really well characterized. So you could very easily, for example, set up a acoustic monitoring system near pig farms, and if you have detection of fruit bat calls, that is, you know, you could use that or something similar as an early warning system for that spatial overlap to say, you know, there is risk of spillover, and put more protection in place. But for something like Ebola, you know, we still don't know what the spillover host is. So it would be very difficult to build an early warning system for that. So I think from an acoustic tech perspective, there's all of the challenges of kind of abundance and actually what metric you would be wanting to measure to feed into an early warning system, and all of the kind of the volume of data that is associated with that, and the cloud-based processing, and sort of how far along the machine learning and pattern recognition, and again, how much training data or how specific that could be to disease hosts. From a disease system perspective, it would be, you know, how well characterized is the spillover that you are trying to have an early warning system for. You know, if it's spillover of disease X, then we have no idea what that looks like. So you could potentially build in something more generic for ecosystem health, or, you know, general degradation of forest, or spatial overlap of particularly high risk, you know, humans with non-human primates, or humans with specific bat populations that are known to host, you know, a variety of high risk viruses. But yeah, I think it would depend on exactly what you're looking for and how specific the system is, how well you understand the ecology to build that in. Sorry, that was a bit of a long answer.
Great, thank you so much.
Wonderful. And I realize that we're pretty much at time, so I'm just going to sneak in this very last question from Kate P. Great talk. Following on to the last question, could you discuss whether you've had any feedback from local communities or governments in integrating passive acoustic monitoring into disease monitoring systems, or what the potential is there? Oh, I think you're muted, Emilia.
Sorry. Classic. So I think that generally positive. I think, as with anything, it depends how well you do your sensitization. So maybe that's not so much an issue in kind of biodiversity and ecology, or maybe it is. But I think that you need to make sure that the communities you're working in and around are appropriately sensitized, or your acoustic monitors are nicked, basically, or they go missing, or they're played with, or they're repurposed for other things. So we've had a few instances of that, but actually working with local collaborators, local governments, sort of ministries of health, I think the ease of kind of, now that this tech is so much more accessible and quite readily user-friendly, I think people are really excited about it, and generally the feedback's been really positive. And there's still a lot of potential for integration into disease monitoring, so we're still mostly seeing it in a biodiversity context. And there's the additional layer of kind of adding adding more into an already overburdened healthcare system. So there's a little bit of difficulty around that. But I think generally positive.
Wonderful. I think that pretty much wraps everything up here. Thank you so much for taking the time to join us and prepare that talk. I think that I speak for the room here that really provided some new perspective for us on how to think about passive acoustic monitoring. Seeing lots of claps in the room, well deserved. Thank you so much. And again, if you need to, if anyone wants to reach out to Emilia and the team, I'm sure their inboxes are open and it goes both ways. Yeah, any parting words here, Emilia?
No, but thank you so much for inviting me. It's nice to pitch to a new audience, and I hope that yeah, I hope there's some interest and intrigue for everyone here.
Wonderful. Thank you all for taking the time to join us, and enjoy your day wherever whatever's remaining of it in your part of the world. Bye everyone.
Thank you.
Thank you. Bye.